Ladies and gentlemen, it's an honor to deliver my talk before this international interdisciplinary research community. My talk is on nanofluidic approach to brain-water metabolism and related issues. It should be observed that our current knowledge of neural circuits can only partially explain the physiology of the brain. To get a complete picture, it is equally important to consider the role of brain-water metabolism and the interstitial fluid that bathes the nerve cells. The interstitial fluid forms external medium for the neural cells and is involved in a number of important processes like non-synaptic intercellular communication, signal transduction, information processing and integration, coordinating response to changes in the external and internal environment of the brain. The interstitial fluid participates in transit of signaling molecules, neurotransmitters, in transfer of substrates, gases, drugs, in elimination of waste products, and in tissue heat exchange. A number of serious neurological conditions arise from or aggravated by the altered brain-water metabolism, like Alzheimer's disease, idiopathic normal pressure hydrocephalus, migraine, traumatic brain injury and stroke, tumor cell migrations, to list a few. A stumbling block in the brain-water metabolism theories is the mechanism of fluid passage through the non-dimensional interstitial space. A dominating opinion in the medical biological community is that interstitial space of irregular tortuous and narrow slates and channels is too narrow to permit any bulk flow. It rather presents a diffusion barrier to fluid movement. Mass transfer events there should be described in terms of diffusion coefficients, gradients, and channel tortuosity. The very non-dimensionality of the interstitial space is used as an argument for the diffusion barrier theory. On the other hand, experimental results demonstrate fast water exchange between capillaries and brain parenchyma. The small and large molecules may move with the same rate in the interstitial space, while, according to the diffusion theory, they should have individual effective diffusion coefficients. There has accumulated enough evidence that rather speaks against diffusion as the only mechanism of fluid movement and mass transfer in the brain. Next, I'd like to demonstrate some figures to illustrate my point. This figure gives an idea about the confinement size distribution of brain water as seen in the sagittal projection. The interstitial space with the interstitial fluid, colored light blue, occupies up to 20% of total brain volume. Going by its characteristic dimensions of 20 to 40 nanometer, it presents nanofluidic domain containing nanoconfined water. On the macro-dimensional side, there is the cerebrospinal fluid filling the ventricular and the subarachnoid macro compartments, colored deep blue. It occupies about 11% of brain volume and contains bulk water. The exchange between the bulk water of the blood and the nanoconfined water of the interstitial space is controlled at the capillary level by aquaporin 4, the black circles around the capillary, as shown here on C. The gaps, colored light blue, present the interstitial space not given to scale. Keeping it to scale here would have turned them into a thin disappearing line. The blood cerebrospinal fluid barriers regulate water flow between bulk water volumes. These are the arachnoid granulations, D, the choroid plexus, B. The brain fluids continue on through compartments varying in size from the nanodimensional to the macro-dimensional ones, with the extended nanocompartments and micro compartments in between. The cerebrospinal fluid is in a constant to-and-fro motion driven by the oscillations of a brain intracranial pressure. Both the cerebrospinal fluid and the interstitial fluid present one functional moiety of freely communicating fluids. This figure presents the mechanism of water exchange between a capillary and the brain parenchyma after the current view prevailing in the medico-biological community. The capillary is in red. The transcapillary water exchange is controlled by aquaporin 4 of the blood-brain barrier, the blue dots along the capillary wall. The movement of water is driven by the osmotic and the hydrostatic pressure gradients. The dashed blue arrows present possible water fluxes. This mechanism essentially originates from the Ernest Starling's model. The brain extracellular space is considered a diffusion barrier, the area colored in gray. The bold blue arrow shows the bulk water flux crossing the diffusion barrier on its way from the periarterial to the perivenular spaces. This after the so-called lymphatic mechanism used to explain waste product removal from the brain, in particular beta amyloid related to the Alzheimer's disease pathogenesis. Incidentally, this bulk water movement is incompatible with the diffusion barrier theory. The following information is meant mainly for the benefit of those members of the medico-biological community who view the brain interstitial space as a diffusion barrier. I'm afraid for those familiar with nanofluidics, it might seem trivial. Nanofluidics deals with the behavior fluids confined to nanosleeves, nanochannels, nanopores, where at least one characteristic dimension is in the nanometer scale, usually from 1 to 100 nanometer. Incidentally, the brain interstitial space falls exactly into this nanometer range. Surface effects in nanoconfined fluids dominate over the bulk water properties, while the fluids exhibit physical behavior not observed in larger structures. A special feature of water movement in nanodimensional spaces manifests itself in significant enhancement of fluid flux due to the surface hydrodynamic slip. Thus, aquaporins, the water-conducting nanopores, exhibit water permeability typically 3 orders of magnitude higher than expected from the classical no-slip from work for the same pore size. From the conventional point of view, this seems counterintuitive and even unsupported when using the Darcy's laws or the no-slip Hagen-Poiseuille's equations, as has been done so far to model fluid movement in the brain. This figure presents the nanofluidic mechanism of brain water metabolism. Contrary to the idea of the brain interstitial space as a diffusion barrier, we view the interstitial space as a nanofluidic domain where fluid movement is governed by the slip-flow principles of nanofluidics. As in the previous figure, the water exchange takes place at the capillary level. Aquaporin 4, the blue dots along the capillary wall, controls water permeability through the blood-brain barrier. The overall fluid transfer between the capillary blood and the interstitial fluid is isosmotic. The pulsatory intracranial pressure presents a driving force behind the fluid exchange between the capillaries and the interstitial space. There are revealed 3 distinctive functional areas along the capillary: the capillary filtration, the capillary oscillating, and the capillary absorption area, CFA, COA, and CRA respectively. The blue arrows show the direction of water fluxes in each area. These figures present an overview of our results on modeling the brain water metabolism carried out after the nanofluidic mechanism. We used a modified phenomenological fluid flux equation based on the Kerim-Kachalsky formalism of irreversible thermodynamics to describe water movement between the capillary blood and the brain interstitial space. In view of time limit, I cannot dwell on this research in detail. I would suggest my free access papers listed by the end of my talk for full information. These figures present modeling of the convective transport of glucose, oxygen, and elimination of carbon dioxide within the neurovascular unit based on the nanofluidic mechanism. It demonstrates from good to high functional capacity of the nanofluidic mechanism as far as mass transfer of the substrate is concerned. Full information on this research could be found in my paper that appeared earlier this year. The nanodimensionality of the interstitial space presents a theoretical watershed. Commonly used in the medico-biological community, the no-slip approach to the interstitial fluid movement and intuitive thinking arising from the bulk water flow experience suggests a diffusion barrier theory and the use of the Fickinean diffusion laws to describe mass transfer events there. An interdisciplinary approach suggests using instead the slip flow principles of nanofluidics to the nanodimensional interstitial space and provides theoretical grounds for the nanofluidic mechanism of brain-water metabolism. The nanofluidic mechanism presents an important missing piece in the jigsaw puzzle of the brain-water metabolism and strongly suggests necessity of paradigm shift from the orthodox diffusion barrier concept to the nanofluidic one. The presented nanofluidic model of brain-water metabolism can be employed in neurobiological research, development of the aquaporin-targeted drug therapy, optimization of the intrathecal drug delivery to tumors and others. Implementation of the new knowledge and its translation into basic and clinical research would open new perspectives in our understanding of brain physiology, pathology, and therapy. It holds a promise of fascinating research along with new challenges. Thank you for your attention.